Now let's take a look at the events occurring in photosystem 1. Just as we saw in photosystem 2, photons of light are hitting the chlorophyll pigment in the antenna complex, and the energy from those photons is being absorbed. The energy gets passed from chlorophyll to chlorophyll to chlorophyll in the antenna complex until it reaches the reaction center. Excited electrons in the reaction center are then transferred to the electron transport chain. In photosystem 1, the electron transport chain is going to do something we haven't seen it do before. It's going to pass down the electrons, but not for the purpose of generating a hydrogen ion gradient. Instead, the electrons will be transferred to the electron acceptor molecule called ferredoxin, and that ferredoxin will then donate electrons to NADP+ to generate NADPH. This is a drawing that's very similar to the drawings we looked at for photosystem 2. Remember, ignore the balance numbers in this drawing. It's not important that you think about whether or not 1 or 2 photons are being absorbed for each electron that's being transferred. What's important to remember is that the photons are hitting the chlorophyll in the antenna complex, and that the energy is then being transferred to the reaction center chlorophyll, which then transfers an electron into the electron transport chain. That electron gets passed down the electron transport chain to an acceptor molecule called ferredoxin. That ferredoxin then passes the electrons on again to NADP+, so it becomes NADPH. So the chlorophyll in the reaction center is being oxidized. The electron transport chain is being reduced. Then the electron transport chain gets oxidized and ferredoxin gets reduced. Then ferredoxin gets oxidized and NADP+ gets reduced to produce NADPH. So we've said that during photosynthesis, during the light-dependent reactions, photosystem 2 and photosystem 1 work together. It's just that photosystem 2 is the first part of the process, and that is followed by what happens in photosystem 1. And that brings us to this diagram, which is known as the Z scheme. It got its name because, of course, the drawing makes this look like the letter Z, sort of, and turned on its side for sure. But you can see where the name came from. What's happening in the diagram is the events of photosystem 2 followed by the events of photosystem 1. We can see ATP produced from the proton motive force in photosystem 2, and we can see NADPH produced during the events of photosystem 1. You can see the splitting of water occurring down here on the left-hand side as well. So the question is, what is it that connects the photosystems? What is it that allows the events of photosystem 2 to sort of trigger the events in photosystem 1? And the answer is a very specific type of molecule called plastocyanin. At the end of photosystem 2, just like at the end of the electron transport chain in cellular respiration, there has to be a molecule there to act as the final electron acceptor. And that molecule is plastocyanin. The electrons that are coming down the electron transport chain in photosystem 2 have to be taken in by something. They have to be received by an electron acceptor molecule. and that is this molecule called plastocyanin. What makes plastocyanin unique is that it turns around and then donates those electrons into photosystem 1. And that's what we can see in the Z scheme. If you look over here, this blue circle represents plastocyanin. Electrons are coming down the electron transport chain in photosystem 2. Their energy is being harvested, the proton gradient is being generated, ATP gets made. But at the end of this electron transport chain, something has to receive those electrons, and that's what plastocyanin does. But what plastocyanin also does is replenish the electrons in the reaction center of photosystem 1 so that these events can keep occurring. And of course, at the end of photosystem 1, the final electron acceptor is NADPH. So if we go back to our sunshine diagram, we've now taken energy from the sun, and through the light-dependent reactions, through the actions of photosystem 2 and photosystem 1, in addition to what happens with water, we've created both ATP and NADPH. Again, the energy, the light energy, has been converted to chemical energy, to potential energy in these molecules. Those two molecules are then going to be used in the process known as the Calvin cycle, along with carbon dioxide gas, to produce the final product of photosynthesis, which is carbohydrate. And remember, in organisms that can do photosynthesis, this carbohydrate is being produced for a reason. It's being produced because these organisms will then use this carbohydrate for cellular respiration. Plants and other photosynthetic organisms are going to produce their own carbohydrate, which they can then turn around and use for cellular respiration to make ATP for energy purposes. So let's talk about the details of the Calvin cycle. The Calvin cycle got its name because it was named after the scientist who outlined the events that occur during that cycle, during that production of carbohydrate. Melvin Calvin was his name, and he was interested in what happens to the carbon dioxide that gets taken in by photosynthetic organisms in the production of glucose. What Calvin discovered was a very interesting molecule, a 5-carbon molecule that no one had discovered before, by the way, which could combine with carbon dioxide and produce two 3-carbon molecules called 3-phosphoglycerate. It's 3-phosphoglycerate that then gets cycled into a set of reactions that make glucose. So what was this previously unknown 5-carbon molecule that Calvin discovered and described? It's called ribulose bisphosphate. Ribulose bisphosphate goes by the abbreviation of RUBP. RUBP is able to combine with carbon dioxide gas and produce two 3-carbon molecules called 3-phosphoglycerate. That 3-phosphoglycerate enters into what's known as the Calvin cycle to produce glucose. There are actually 3 distinct stages or phases of the Calvin cycle, and all of these are occurring in the chloroplast in the stroma. The 3 phases of the Calvin cycle are called fixation, reduction, and regeneration. Let's take a look at each one. During the fixation phase, carbon dioxide gas is fixed and that word means transformed into a more usable form for the organism. It's fixed by attaching it to RUBP. This produces those two molecules of 3-phosphoglycerate that I mentioned earlier. In the reduction phase, the 3-phosphoglycerate is combined with the products of the light reactions to produce a molecule called G3P, which stands for glyceraldehyde 3-phosphate. So 3-phosphoglycerate is combined with ATP and NADPH to produce G3P, glyceraldehyde 3-phosphate. Now, during the regeneration phase, some of the G3P will be used to build glucose. And remember, that glucose can then be used to build other carbohydrate that the plant might need, or it can be used directly for cellular respiration. The remainder of the G3P that's produced during the reduction phase is going to be used to regenerate that critical 5-carbon molecule called RUBP. The RUBP will then re-enter the fixation phase. This is why this process is a cycle. It keeps going around and around because at the end of the regeneration phase, we make the molecule that's needed to start the whole thing over again. On this slide, we can see another depiction of the Calvin cycle, broken down into its 3 stages. Remember, all 3 phases of the Calvin cycle are occurring in the stroma, and that's the space that you can see outside of all of these stacks of grana in this drawing of a chloroplast. During fixation, RUBP is being combined with carbon dioxide to make 3-phosphoglycerate. During reduction, the 3-phosphoglycerate is being combined with ATP and NADPH to make G3P. Some of that G3P goes on to produce glucose. The rest of the G3P is used for the regeneration phase, where it is converted with ATP into the 5-carbon molecule RUBP, and the RUBP is then fed back into the fixation phase. On this slide, we can see the Calvin cycle drawn as a cycle, as a circular process. At the top, we can see the carbon dioxide coming in to the cycle and combining with the 5-carbon RUBP molecule. The 5 red circles represent 5 carbons. 5 carbons from RUBP, 1 carbon in CO2, and we get out of that 2 3-carbon molecules, 3-phosphoglycerate. As 3-phosphoglycerate enters the reduction phase, and remember in that phase it will be used to produce G3P, this is where we see the ATP used, it'll be broken down, and the NADPH used, it will become oxidized, and G3P will be the product. Some of that G3P is then used to build glucose, and of course the glucose then can be used to build other carbohydrate, or it can be fed directly into cellular respiration. The remainder of the G3P molecule is then used to build RUBP with the energy from ATP. The RUBP will then be once again entered into the fixation phase, and the cycle continues. Now, one other thing to note about the Calvin cycle. The Calvin cycle is telling us what the products of the light-dependent reactions are being used for. the ATP and the NADPH that's produced during the light-dependent reactions get used by that photosynthetic cell to reduce carbon dioxide gas and produce glucose. That first fixation reaction where carbon dioxide combines with RUBP, it's an unusual reaction. RUBP is an unusual molecule. So there's a particular enzyme that does this work. And this enzyme is called ribulose 1,5-bisphosphate. It goes by sort of a nickname of Rubisco. On the slide down at the bottom, you can see a depiction of the three-dimensional structure of Rubisco. It shows us that, like many enzymes, Rubisco is enormous. It's an enormous protein. The other thing that you can't really tell from this depiction, but we know from other studies, is that Rubisco actually has a cube shape. It literally has the shape of a cube. Very unusual. The other interesting thing about Rubisco is, compared to other enzymes, Rubisco is amazingly slow at catalyzing this reaction. It can only perform about 3 reactions per second. Now, I know that sounds fast, but remember, during things like DNA replication, enzymes are capable of, particularly the DNA polymerase enzyme, capable of adding 100 or so bases per second to the growing DNA strand. So this enzyme, with 3 reactions per second, is pretty slow. Very, very strange, very interesting. The other thing to know about Rubisco is this. It turns out that Rubisco is the most abundant enzyme on the planet. And that's because it is found in every single photosynthetic cell on the planet. Something to think about.